The capacity to acquire large amounts of data from calcium imaging experiments has drastically improved, especially with tissue imaging experiments. Our protocols describe appropriate analysis techniques to quantify and describe these datasets. Our protocols generate a range of spatial, temporal, and intensity values that are intrinsic to calcium signaling in entire tissues, and this can be lost in more rudimentary base analysis.
One hour before the imaging, transfer a small intestine harvested from a mouse with interstitial cells of Ca2, or ICC, that specifically express a genetically encoded calcium indicator to the stage of a spinning disc confocal microscope and continuously perfuse the tissue with 37 degrees Celsius Krebs-Ringer bicarbonate, or KRB, solution. Using a 60 to 100x magnification, locate the stellate-shaped myenteric plexus ICC, or ICC-MY, between the circular and longitudinal smooth muscle layers of the small intestine. Then, locate the spindle-shaped deep muscular plexus ICC, or ICC-DMP, in a single plane between the circular smooth muscle layer and the submucosal plexus, and save the movies as a stack of TIFF images. To create spatio-temporal maps, or STMs, of the ICC-DMP calcium activity, open the image stack in ImageJ and click Image, Type, and 32-bit to modify the image quality. To create a line scan, right-click on the Line Selection tool and select Segmented Line. Using